A very good morning all of you. Welcome for the second session of the basic cancer biology online video lecture. So I am Dr. Ashok Kumar Fondarangan, Associate Professor working in School of Life Sciences, PS Abdur Rahman, Crescent Institute of Science and Technology, Vandalur, Chennai. So in the previous lecture we discussed about the basics of cancer, the definition for cancer, what are the classifications are there, benign tumors, malignant tumors, and what are the causes, the history behind the naming of tumor. All these things we discussed in the previous part of the video. And this part of the video we're going to discuss about some crucial part of cancer. We're going to discuss the cancer family of genes called as oncogenes and tumor suppressor genes. So generally oncogenes and tumor suppressor genes are two ends. Oncogene which is promote cell growth and which is highly upregulated in the tumors. It also enhances the motility of the cell. But in case of tumor suppressor, it is totally opposite. In contrast to oncogene, it suppresses cell growth, it acts as a regulator, and its level were always downregulated in the tumor cells. So both play opposite role. So the name itself implies tumor suppressor is they suppress the tumor. First we will see in detail about oncogenes. So the term oncogene was coined in the year 1969 by Hubner and Todaro. So these genes have the potential to induce cancer. That's why they called as pro-oncogenes. They can cause cancer. So the transformed healthy cells cause them to gain the hallmark of cancer. So the hallmarks of cancer we will discuss later part of this video. So this oncogenes first discovered in virus, later in the cells. So these viruses inject these genes into the human cells, which stays in the human later. So SRC was the first oncogene which is discovered before people understand what is oncogene really. So first I told you the Ross who worked in the Rockefeller Institute of New York. He is the first person who identified this Ross sarcoma virus which induced virus. So in the year 1911. So what he did was he did a simple experiment. He took a cell-free extract from chicken tumors which cause new tumors when they injected to healthy chickens. So that experiment he found that this SRC oncogenic retrovirus, which is the reason for inducing Ross sarcoma, which causes sarcoma in the chicken. So some terminologies which is discussed in the right side as Ross sarcoma, which is a form of cancer which infects chicken, and Ross sarcoma virus RSV, which is a retrovirus. A retrovirus means a virus which contains RNA as a genetic material instead of DNA. So it generates which is a retrovirus which generates a cDNA that inserts into the chicken's DNA. So VSARC was virus SRC. That means the gene which gets expressed by Ross sarcoma virus. So this names included Ross because the Ross was discovered all these events. So some of the other viral oncogenes which were described in this slide. So Ross sarcoma was SRC, named as SRC. Blazon leukemia, ABL, which is abbreviated as ABL. Avian erythroblastosis, ERBB. Medonophilin sarcoma, abbreviated as FMS. HS filin, abbreviated as KIT. Moraine sarcoma 3611 was abbreviated as RAF, RAF. Simian sarcoma, which is abbreviated as SIS, Harvey sarcoma, which is abbreviated as HRAS. So these are the other viral oncogenes which is identified so far. So how this SRC, which is found in other human tumors, SRC expression is upregulated in other tumors also, not only in the Ross sarcoma. Other tumors such as breast cancer, pancreatic cancer, ovarian cancer, head and neck cancer, lung cancer, gastric cancer, and in colorectal cancer. All these cancer types express SRC oncogene. So how these proto-oncogenes were activated? Normally you need activation of this proto-oncogene to cause cancer. So proto-oncogenes generally they are tightly regulated in the healthy cells. They remain silent. When these three events such as mutation, gene amplification and chromosomal translocations, these three events can activate the proto-oncogenes. So another class of cancer genes were tumour suppressor gene. So as I mentioned earlier, they suppress growth in the healthy cells. They act as a brake. The loss of tumour suppressor gene may also cause cancer. Not only activation of proto-oncogene can cause it. On the other end, when there is a loss of tumour suppressor gene, that also can cause cancer. That's why cancer is defined as either gain of oncogene or loss of tumour suppressor gene. So how these tumour suppressor gene were inactivated? What is the mechanism involved in it? So these are the list denotes which causes the tumour suppressor gene inactivation. Gene deletion, either the gene was deleted, direct mutation, that mutation change in the DNA structure that causes tumour suppressor gene inactivation, loss of heterozygosity. So loss of heterozygosity it means two copies of DNA vary in this tumour suppressor gene. Epigenetic silencing, there is methylation in the promoter, that also causes the inactivation of tumour suppressor gene. So here I gave some list of tumour suppressor gene which is already identified. P53, one of the very important tumour suppressor gene which is there in the which is identified. Adenomatous polyposis coli, APC. This frequent mutations, around 80% of the APC mutations were the reason for the development of colorectal cancer. BRCA1 is also very important tumour suppressor gene. CYLD, neurofibromin1, CDKN1B. All these are all important tumour suppressor genes. So Douglas Annan and Robert A. Weinberg, these two scientists, they proposed 6 common hallmarks of cancer. So all cancers have share these 6 common traits that govern the transformation of normal cells to malignant tumor cells or cancer cells. Let us see what are those 6 hallmarks of cancer. Evading apoptosis, self-sufficiency in growth signals, insensitivity to anti-growth signals, sustained angiogenesis, limitless replicative potential, tissue invasion and metastasis. We are going to see each of these hallmarks in detail. First one is self-sufficiency in growth signals. So these cancer cells do not need any kind of stimulation from the external signals to multiply. So they multiply on their own. There is no such stop signals are there. It can have anytime it can replicate, anytime it can proliferate on its own. So it no need any external stimuli to start the multiplication. Their insensitivity to anti-growth signals. In the normal cells, they respond to these anti-growth signals and stop the proliferation. But cancer cells generally they resist to this growth preventing signals. That is the second hallmark of cancer. So the third one is evating of programmed cell death. You know very well apoptosis is the programmed cell death where the cell is cleared from this from the system through a proper mechanism to not affect the neighboring cell. Okay, the cell used to die in a programmed way. So these cancer cells, they have a mechanism to escape or bypass the apoptosis. So as I said earlier, cancer is uncontrolled growth of cells. So they have this limitless replicative potential. Non-cancer cells die after certain number of divisions, but cancer cells escape from these limits, are afferently capable of indefinite growth and division. That is called as immortality. But these immortal cells have damaged chromosomes which can become cancerous. Another important hallmark is developing blood vessels. Angiogenesis is a process where new blood vessels are formed from the existing blood vessels. So these cancer cells appear to able to kickstart this process, ensuring that such cells receive a continuous supply of oxygen and nutrients for their growth. So another important hallmark is tumor invasion and metastasis. This we discussed in the first part of the video. So these cancer cells break away from their primary site of tumor, and they escape and they enter into the bloodstream. They invade the surrounding tissue and start spread. Sometimes it travel to the distant body parts. So the deregulated metabolism is another type of hallmark. So most of these cancer cells use abnormal metabolic pathway to generate energy. So Otto Warburg, he proposed an hypothesis in the year 1914. So what he said was cancer cells acquire energy by means of glycolysis alone. So he found that glycolysis was 10 times higher than the normal cells. The reason what he found was the tumor is spherical, so the inner layer of the tumor, they are hypoxic, they experience hypoxic condition, so they cannot able to rely on oxidative phosphorylation. Hence it is totally rely on glycolysis to produce ATP. He also found that this hypoxia inducible factor 1 alpha gene expression was higher in tumor. The role of E4 alpha is to release the glycolytic enzymes to have more glycolysis. Based on this hypothesis, a technique is identified that is called PET scan, positron emission tomography. So evading of the immune system, cancer cells appear to be invisible to the body's immune system. So it escapes from the immune system, that's why it can able to grow. And another important hallmark is genomic instability. Cancer cells generally have severe chromosomal abnormality which worsens as the disease progresses. And inflammation. Local chronic inflammation may induce many types of cancer. Okay, so in this part of the video we're going to see what are the cancer diagnostic methods are there and how they are diagnosing the cancer. So in this slide you see the cancer diagnosis were majorly categorized into imaging techniques, endoscopic techniques, biomarker based methods and other methods. So first, the imaging techniques involved X-rays, CT scan, isotope scan, ultrasound scan, bone scan, MRI and PET scanning. And the second method is endoscopic method. So this endoscopic method is especially for the diagnosis of cancer which is there in the internal part of the organ. So since it is an invasive method, that's why people used to go for imaging techniques. Imaging techniques means it don't affect much when you go for imaging techniques, but where endoscopic method is little invasive, that's why people used to go for imaging techniques. The third one is biomarker based methods. So in this method you have immunohistochemistry, detection of cancer, flow cytometric methods are there, Western blots, genomic and proteomic approaches where you can go for next generation sequencing. The others involved biopsy, screening test, both blood, serum, WBC count, the white cell count, erythrocyte sedimentation rate, that is ESR, and serum biochemistry. So in the blood serum test you have specific cancer biomarkers are there, that is carcinoembryogenic antigen and alpha fetoprotein. But unfortunately these you can detect if in your blood the carcinoembryogenic antigen is detected high level than the normal, so it implies only the cancer is there, but you don't know which organ is affected by the cancer, where the cancer is there. To locate the cancer is very, very important. So there comes the imaging techniques help you. So the X-ray, CT scan and MRI, PET scan, all these things will help you to locate where your tumor resides. So we will see how these things were done one by one. So this is the X-ray image of lung cancer. You see a person who affected by lung cancer, he undergo X-ray. This is very primitive technique, X-ray imaging, but still it can detect the cancer specifically in the lung, on the right side of the lung, which was affected by, invaded by a good big amount of cancer was seen. You can see that cancer. And the second method is PET-CT method. So I told you Warburg, based on the Warburg hypothesis, this method was developed. So what they do, they take the analog of glucose, which is detected, which is attached with some specific fluorescent probes. So they will give it to the patient. So the patient will ingest that glucose analog. After he injected, the body try to metabolize those glucose analog. So wherever the cancer is there, the glycolysis will be higher, right? So what will do this glucose analog? It will accumulate where the tumor is there. So when after 15 minutes, when you undergo this positron emission tomography, so it will show this fluorescence is there attached with that glucose analog. So it will show where the site of the tumor and the size of the tumor. You can see the cancer was located in this person. The liver was affected by cancer, and in the lung the cancer was detected in the left side, the November 16 image. So the next one is MRI. MRI is one of the advanced method to detect tumors. You see the brain tumor was detected by MRI scanning. So I told you already the endoscopic methods are generally employed to use to check or diagnose the cancers of internal organs. So what they do means the procedure is little bit tough. So for example, if a person is affected by colon or stomach cancer, a small tube which is not more than 1 to 2 millimeter size, at the end of this tube they attached the camera. So they inserted through your mouth to the stomach, where to check any signs of tumor is there. So it will lively, the clinician or physician can see whether any tumor signs are there in the stomach. So similarly they will go for colon, larynx, pharynx, bronchial, all these examination they can do by means of these endoscopic methods. It's very simple, but it is invasive method. Patients will feel little uncomfortable when they do endoscopic methods. Here I gave two images. One is the tumor was detected in the colon using colonoscopy, and on the right side image the gastric adenocarcinoma was detected in the endoscopic method. This is how the tumor will look like. So another method in the others was biopsy. So biopsy will give you specific information whether the suspected organ, whether you have tumor or not. So what they do is a small hole was wherever the target organ, they will have a small hole and pinch a small piece of tissue, and that tissue they will use for histological and immunohistochemical methods to detect whether this particular organ is affected by cancer or not. So this is histological detection of tumor. So in this picture, left adenoma, you see the normal mucosa which is there. What they do to do histology means a small piece of tissue will be embedded in the paraffin. Then they cut into small slices like 5 micrometer sections, and then they stained with methoxylin and eosin. So after that, when you view under microscope, you can locate specifically the normal mucosa and tumor you can detect under microscope. So this is another specific method, others diagnostic method is immunohistochemistry method. So these sections were stained with the PCNA, that is proliferating cell nuclear antigen, which shows the highly proliferating cells. So these sections were stained with this PCNA antibody, and you see the normal mucosa which has very not that intense brown color, and you see the tumor region is highly intense brown color. That's because of the tumor contain high proliferating cells. Okay, we come to the last part of the video lecture. What are the treatment options are available to treat cancer? So the list is given here: surgery, radiation therapy, chemotherapy, immunotherapy, targeted therapy, hormone therapy, and precision medicine. So we will see one by one in detail. First we are going to see about surgery. So surgery is the first option for cancer treatment. After a patient is diagnosed with cancer, the doctor will recommend for surgically remove the tumor. Surgery means nothing but it's a procedure which a surgeon removes the cancer from your body. Surgery of tumor doesn't mean that the tumor is completely eliminated. So it gives a first line of treatment where your tumor, the big chunk of piece will be removed and thrown away. So secondary treatment after surgery, the physician may go for radiation therapy. So where the high dose of radiation is passed to the cancer cell to shrink the tumor. This is an alternative treatment method for over surgery. So there are a lot of side effects are there in radiation therapy too. So the next one is chemotherapy. Chemotherapy is nothing but doctor will use drugs, chemotherapeutic drugs to kill cancer cells. So these drugs destroy cancer cells that may remain after treatment with surgery or radiation therapy. This is called adjuvant therapy or additional therapy. Kill cancer cells that have returned or spread to other parts of your body. So here in this table I have given a list of chemotherapeutic drugs which is used for different cancer types. For example, breast cancer, they use cyclophosphamide, 5-fluorouracil, doxorubicin, cyclophosphamide. All these combinations they will use. Likewise for each cancer, for example, Hodgkin disease, they use mustine, vinscretine, doxorubicin, bleomycin, blemdastin. Germ cell tumor, stomach cancer, they use epirubicin, cisplatin, 5-fluorouracil. So for colorectal cancer, specifically they use 5-fluorouracil, folic acid and oxaliplatin. So likewise, the combinations of these chemotherapeutic drugs were used based on the cancer type. So another problem is there in chemotherapy, that is, chemotherapy can cause side effects. So chemotherapy not only kills the fast growing cells, but it also kills or slows the growth of healthy normal cells. So the examples are cells that line your mouth and intestine, those are cause your hair to grow. Damage to healthy cells may cause side effects such as mouth sores, nausea and hair loss. Due to the chemotherapy, the hair loss is very common side effect. Side effects often get better or go away after you finish the chemotherapy. So the next is the recent development that happens in the field of immunotherapy to treat cancer. So immunotherapy is a type of cancer treatment that helps your immune system to fight against cancer. The immune system helps your body fight infections and other diseases too. It is made up of white blood cells and organs and tissues of the lymphatic system. Several types of immunotherapy is classified to treat cancer, that is immune checkpoint inhibitors, T-cell transfer therapy, monoclonal antibodies, treatment vaccines, immune system modulators. Sorry for the spelling mistake there. So first we'll see the immune checkpoint inhibitors. So what are these immune checkpoint inhibitors means? Which are drugs that block immune system checkpoints. These checkpoints are normal part of the immune system and keep immune responses from being too strong. By blocking them, these drugs allow the immune cells to respond more strongly to the cancer. Especially they used to target PD-L1 immune checkpoint inhibitor to treat various type of cancer. The next method is T-cell transfer therapy. So T-cell transfer therapy, which is treatment that boosts a natural ability of your T-cell to fight against cancer. So what they do means, doctors, they isolate a tumor, a part of tumor, and they will check which type of cancer cell, the immune cell which is highly present in the tumor, which resists the tumor cell. So they will grow in vitro condition in the laboratory. Then they multiply the immune cells, and once it is multiplied in good numbers, they will again inject to the patient, cancer patient. These immune cells will go and inhibit the tumor. T-cell therapy may also called as adaptive cell therapy or adaptive immunotherapy or immune cell therapy. The clinical trials are going on in this area. Next one is treatment vaccines. So which work against cancer by boosting your immune system response to cancer cells. It's similar like viral and bacterial diseases. They use vaccines to treat cancer. So the example is HPV vaccine is available in the market now. So you can also use it to prevent cervical cancer. The next important part is monoclonal antibodies. So this monoclonal antibodies, which immune system proteins created in the lab from a single clone of cells, which is designed to bind to specific targets of cancer cells. Some monoclonal antibodies mark cancer cells so that they'll be better seen and destroyed by the immune system. So simply marking of these, using these antibodies to mark the cell, which will be recognized by the immune system and destroyed. Such monoclonal antibodies are type of immunotherapy. Monoclonal antibodies may also called as therapeutic antibodies. The examples I have given two here, bevacizumab and cetuximab. So bevacizumab actually targets VEGF and cetuximab targets EGFR. So next topic is targeted therapy. So this is a type of therapy in the cancer treatment that targets protein that control how a cancer cells grow, divide and spread. Specific type of cancer, a specific type of protein will be upregulated. So targeting that particular protein by means of a small molecule inhibitor or a monoclonal antibody, you can control the tumor, or you can sensitize these tumor cells, and if you provide a proper chemotherapeutic drug, so this chemotherapeutic drug will induce apoptosis because the cell is already sensitized by a specific targeted small molecule inhibitor or monoclonal antibody. So as researchers learn more about the DNA changes and proteins that drive cancer, they are better able to design promising treatments that target these proteins. So two categories of targeted therapy I have given here. One is the small molecule inhibitors. Specifically, you can design small molecules to target specific protein, or monoclonal antibodies also you can use. So hormonal therapy is one another method to treat cancer. So hormonal therapy is a cancer treatment that slows or stops the growth of cancer that uses hormones to grow. So far we have seen all those treatment methods currently available to treat cancer. So these are the references I used to make this video. Thank you so much for following up. I hope you understand some of the basic cancer biology. If you have any questions, please, my email ID is there in the first part of the video, so you can send any questions to me. I'll be very happy to answer all the questions to you. Thank you. Good luck for your future.